and other research today. And I will let Mark take over now.
Thanks, Eve. Hopefully everyone can hear me okay and see the slides. So yeah, as Eve said, I'm going to be talking about adaptive study designs. So I guess we should start with the definition. So in an adaptive study design, some aspects of the study are modified in light of the accumulating data. So you look at your data and something about your study changes. So again, this is a fixed study design. So you plan how your study is going to work. You start the study, you collect your data, and then you stop collecting data, you analyze it, and you draw some conclusions. In an adaptive study design, we have some extra steps. So while you're collecting the data, you can kind of stop and analyze it. And then you can make a decision. You can decide that you want to modify the study in some way. You can decide that you continue without making any changes and go back to collecting data. Or you might make the decision to finish the study there and then analyze it and draw your conclusions. And this can be kind of iterative. So you might modify your study the first time you look at it, keep collecting some data, and then decide not to modify it the next time, and then do another interim analysis. And you make these decisions based on what you're seeing in that data that you've collected so far. So I'm just going to give an example of probably the simplest adaptive study design you can have, which is early stopping. And this is a very adaptive design. So this was published not so long ago. Someone said their sample size justification was that they continuously increased the number of animals included in the study until they got statistical significance to support their conclusions. So this is very adaptive because they're kind of checking the results after every sample is added and deciding whether to continue or stop at that point. So I'm just going to give an example of how this might work in practice. So I'll start just by outlining what this graph is going to show you. So I'm going to talk hypothetically about an experiment we're doing where the truth is that there's a difference of 0.5 in between, let's say, two groups. And what we're going to do is we're going to increase the sample size, and after each pair of observations, one in each group, we're going to estimate the difference from all of our data up to that point, and update our estimate. And the truth of that 0.5 is just marked with the dotted line here. So let's see how that looks. So this is our first kind of hypothetical experiment. You can see kind of at the start, when we've got small sample size, our estimate of the difference can move around a fair bit. But then as our sample size increases, it gets closer and closer to the truth of 0.5. That's a second hypothetical result from this experiment. A third one, I think there's 20 lines there. You can see they're all doing kind of a similar thing. They might start pretty volatile and then they settle down and get close to the truth as we increase the sample size. And that's, I think, 500 experiments. And again, we're seeing the same pattern there. So that is good. But now we want to apply this adaptive study design. So what we're going to do is we're going to set an alpha level of 0.05, which is what a lot of people use. We're going to compute a p-value at every sample size. So after each new set of observations, we're going to compute the p-value. And if our p-value is less than this alpha level of 0.05, we're going to stop the study, just like our design told us to do. And that that corresponds to these thinner lines here, in the sense that if our estimate gets far enough away from zero, we get a small p-value. And so if our estimate goes outside these lines at any time, that's when we would get a p-value less than 0.05 and we would stop the study. So our first hypothetical result is this green line here. And you can see the green line kind of wobbles around a bit. And then after, I don't know, about 50 or so observations, it crosses that boundary and we stop the study and we declare that we've got a significant result. We've got a second hypothetical result. You can hardly see here because it starts off quite high and it only takes a few observations before you jump outside that boundary. Third one does a similar thing. When we get 20, we can see a bit more variability, but because these boundaries kind of move far away from the truth of 0.5 kind of reasonably early, all of our experiments are stopping quite early. They're stopping, within 125 sample size as they cross that threshold of significance. And that's a full set of 500 hypothetical results. You can see that none of them, well, I think there's one that gets beyond like 125 sample size. So that's good. Our power is 100%, and the median sample size is 13. So half the time we don't need any more than 13 samples before we get significance. That's great. What if our true effect size was smaller? So the difference between the groups is now only 0.2. So this dotted line has kind of moved a bit closer to 0. So I guess we'd expect to see the hypothetical experiments kind of stay within these boundaries for a bit longer. And that's what happens. So this first one goes for quite a while before it gets outside the boundary. The second one stops kind of quickly. Third one, 100-ish observations before we stop. And there's a bunch and a bunch more. And we can see they're doing a similar thing. So they can start off quite volatile. And as they try to get close to this truth, they're more likely to fall outside our boundaries. And hey, this is pretty good. We've still got 100% power. Now our median sample size is 63. But that's all right. We're able to detect this difference every single time by running this experiment. What if we make the difference really, really small? If we make it 0.05, let's see what happens. Our first experiment stops, after about 200 samples. Our second one is in red because it doesn't stop before 1,000, but we never said we're going to stop at 1,000. We're just going to keep going until we get significant results. And it turns out that one actually does go outside the boundary. So does that one. And when there's 1,000 lines there, you can't see very much. But I can tell you that the power is still 100%. All of these hypothetical experiments eventually declare a significant difference between the groups. It takes obviously a bit longer here, but our median sample size is still 428. So we are getting there eventually most of the time. So this kind of seems pretty good, right? Normally statisticians will tell you, if the effect size is smaller, your power is going to reduce, right? But here we've got an experimental design where our power is 100% no matter what the effect size is. And that's pretty good. That seems like a free lunch. But you want to look out for banana peels. And I might show you one of those now. So what if there is no effect whatsoever? So the difference between the groups is 0. We're going to keep our same threshold of 0.05 p-value. We can see our very first hypothetical experiment stops at about 200 samples. Our second one gets beyond 1,000 without reaching significance. Third one does the same. But a fair chunk of our first 20 are the green lines there, and they stop before they get to 1,000 and they declare a significant difference. And again, you can't see a lot with 1,000, but you can see that there are some green lines there that are stopping before the sample size gets to 1,000. In actual fact, you can show that this experiment also has a power of 100%, and the median sample size is around 500. So half the time we're able to declare a significant difference before we get to 517 samples. What's the problem with this? This is not really power anymore. This is type 1 error, right? Because there's no difference between the groups, and we're making a mistake every single time we declare that there is a significant difference. So that doesn't seem like a good idea. But you also might say that's not really practical, right? I'm telling you you've got 100% power, but that's only under the kind of idea that you could continue this experiment on for as long as necessary to get significance eventually. And sometimes that might take you, way longer or cost way more money than you've actually got. So let's talk about a more practical example. So let's say we've gone to a statistician and they've told us that we need 85 samples to get 90% power in this situation where the true difference is 0.5. And so now I've compressed these two scenarios. So this is when the true difference is 0.5, 500 example studies. And on the right is when the true difference is 0. So again, 500 example studies with a sample size of 85, but we're checking after every new observation for significance. You can see this one looks pretty good. We're finding a significant difference a fair amount of the time before we get to 85 samples. And on the right-hand side, we don't want to find a significant difference because there isn't one. But we can see a fair number of green lines here, which means that we are stopping early and saying that we have found a difference. So the left-hand side, we've actually got 97% power. So 97% of our studies are finding a significant difference. And the average sample size is actually about 22. So we found a bit of a cheat code here, right? A statistician said you have 90% power with 85 samples. We've actually bumped that up to 97%, and we've bumped the average sample size down from 85 to 22. So that's pretty good. But there's no such thing as a free lunch. If we look on the right-hand side, actually 37% of these hypothetical studies have found a significant difference. So that's making a type 1 error. So that's way higher than the alpha level that we set. And that's probably way higher than people would find acceptable as a type 1 error rate. So it kind of seems like you shouldn't look after every observation. That's probably what's contributing to that type 1 error. So instead, let's say we're able to resist looking after every observation, and we're just going to look once at halfway and then again at the finish. So I don't know how well you can see these, but instead of the thin lines now, we've got these crosses at halfway and at the end. And if at those time points, our estimate is outside of them, we're going to declare a significant difference. And if it's inside them, we're going to declare no difference. And if it's outside of them at the halfway point, we're going to stop there and not collect any more data. So again, this is just a couple of hypothetical studies under each of these scenarios. You can see this one doesn't reach significance at halfway, but it does by the end. So that's all right. We didn't get to stop early, but we got the right result. And this one in the null scenario isn't significant at either time point, so that's cool too, because we shouldn't be declaring that there is a difference between the groups there. Our second one in the first scenario actually is outside our boundaries at halfway, so that's good as well because we found the significant difference and we've managed to stop early, so we don't have to go all the way to 85. And in the null scenario, we've got the right result again here. Third one, on the left-hand side, it is just outside the boundary at halfway, so that's good. And we've got the right result in the second case too. So this is looking pretty good. And if we roll that forward to 20 hypothetical studies, you can see there's just one red one here on the left-hand side. So that's a situation where there is a difference, but we didn't find it to be significant at halfway or full time. And you can see there's a bunch up here where we were able to declare a significant difference, at halfway at that first check. So that's looking good. And in the null scenario, you can see it looks like there's just two cases where we declared significance, one of them at the end of the 85 and one of them at halfway. So this is looking all right. But if we roll that forward to a full set of 500 kind of hypothetical results, there's a decent bunch that don't declare significance in the first scenario. That's not too bad because that's just a lack of power. We already thought we only had 90% power anyway. And in the null scenario, you can see some green ones that stop at halfway and some green ones that stop, that declare a significant difference at the end of the study, and those are type 1 errors. So our power is actually 93%. So again, we're happy with that because we thought we only had 90% power. And the mean sample size is 57.7. So it's obviously larger than it was before because we're not able to stop before we get to 43. But we stopped early 65% of the time. So we're doing much better than having to recruit 85 samples on average. But the downside, again, is that our type 1 error rate is higher than we'd kind of nominally tried to set it at. So this should really be 5%. This is just kind of an estimation error because I just did 500 samples, but you would stop early 5% of the time because that's what our alpha level is. And you'll also declare a difference in some cases that went the full way. So we're going to end up with a type 1 error rate that's larger than our alpha level. So that is not good. What's the solution? Well, the solution is to say that if we want this total to be 5%, then we need to split it up between the studies that might stop early and the studies that might declare a difference at the end. And one method to do that is to use a spending function to distribute how your type 1 error goes at the interim and the final analysis. So there's an infinite number of ways you can do this. Here's a bunch of ones with names. And some of them have like parameters to say, I want to spend more of my type 1 error rate early or later in the study. But yeah, there's in this left-hand column here, or the middle column, this is the p-value that you would set as your threshold, or the alpha level that you'd set as your threshold at the interim check. This is the alpha level that you would set at the final analysis, if we're just doing one interim analysis. And this spend thing here is like the the additional amount of type 1 errors that you would make at the second analysis. So you'll notice that the spend plus the interim alpha is equal to the 0.05 that we want. So there's lots of different options here. So O'Brien Fleming sets quite a strict threshold at the initial analysis. So you'd have to get a p less than 0.005 to declare a difference. And then the final p-value is pretty close to 0.05 because you haven't made many errors early on, so you can afford to make them later. The Pocock sets the same threshold at both analyses. And that means that you are spending kind of more alpha early on and then less later. So you're more likely to stop early, I guess. And I guess you'll be all right with that. And then there's a bunch of other ones, yeah. And you can see as you change this parameter here, it goes from quite a strict interim threshold to quite a liberal one. And the kind of the trade-off is then in the threshold that you use at the final analysis. So let's just have a look at one of those. So I've chosen the Pocock boundaries, which is where you set the same threshold level at your interim and your final analysis, and that's 0.0294. So that corresponds to these cut points or these thresholds here with the little X's. And I won't go through these one by one, but this is what happens here. And we found that we've got 90% power and an average sample size of 61.5. So this is pretty good, right? So instead of doing a study with 85 samples where we had 90% power, now we've got a study which might have 85 samples, but it's got a 62% chance of stopping after only 43 samples. So we've saved a lot of kind of resources a lot of the time for the same amount of power. And do we have the banana peel from before? No, we don't. Because we set a stricter threshold at our interim and our final analysis, we make a type 1 error less often, and it ends up adding up to that total of 5%. So this kind of is a free lunch in some sense. We've got the same amount of power. We've got a lower expected sample size, and we've got the same type 1 error rate. Bit that's not free about that lunch is that I guess you just got to think about it first before you run your study. One thing to think about as well is about your estimates at the end of the study. So if you think that the truth is somewhere, and in this case our truth is 0.5, right? Our data can kind of randomly vary around that truth, right? And the issue with having stopping rules involved is that if our data is kind of unusually extreme, unusually high compared to the truth, we're more likely to stop. So if you only look at studies that stopped early, it's kind of going to be biased higher than the truth. And on the flip side, if your study doesn't meet the threshold early on, that kind of means that it might be kind of randomly lower than the actual truth, or kind of unusually close to zero. And that means you might be underestimating the truth if you just look at the studies that continued on to the final analysis. And so that's what this graph is showing here. So the black curve is kind of the distribution of estimates if we just use a fixed design with 85 observations. The green curve is if we have an interim analysis and we decide to stop early. So you can see that the average result from the studies that stopped early is larger than 0.5. And the red curve is if we look at the studies that didn't stop early and went to the went to the end. And even though, 90% of those are still declaring a significant difference, we can see that they're underestimating the truth. Because to be able to make it to the end, they had to have a small enough estimate at that interim time point. Am I going for time? Okay. So one thing again to think about when you're doing the analysis is whether you need to adjust your estimates for this conditional bias. If your study stops early, you kind of know that it's more likely to be biased upwards. Should you, take a bit off your estimate to account for that? All right. So one other kind of smart thing we can do is stop for futility. So we can say halfway through that if it's not looking like we're going to get a significant difference by the end, we might stop and not waste our time. So I've just included some thresholds here in the circles. So what's going to happen here is that if at halfway our difference between the groups is so small that it's looking like it's not going to be significant by the end, we're going to stop and just say there's no significant difference at halfway. And we're making a binary choice at the final time point. So those thresholds are at the same place. So we can see what happens here. Our first one on the left stops early, but we're declaring a difference. Our one on the right isn't between those thresholds in the middle. So we think there's a chance we still could see a significant difference, but when we reach the end, we don't see a significant difference. We keep going forwards. We can see, I think this is our third one on the right. It's actually dipped between those little gates at halfway. So we've declared, we've got such a small difference between the groups that we're not going to get there. We're not going to see a significant difference at the end. So we're going to stop early. And that is the right decision in that case. We want to stop early for futility under H0 when there is no real difference. And again, we keep doing that. And this is what we see. We can see that when there is a difference, there's a very small chance that we'll actually get futility at the interim analysis sometimes. So some of the red lines that stopped at halfway. And this tiny little white space here, if you can see it, this is like the efficiency gain in this case. So under H0, when there's no true difference, some of our hypothetical studies kind of go between those gates at halfway, and we are able to stop early and declare that there's no difference without having to go all the way to, oh, this is not 85. This is like, for some reason, the software wouldn't give me a sample size of 85. So this is closer to 100. So here, the power is still 90%. Our mean sample size is 66.6, so that's pretty good. 61% stopped early for efficacy, and 2% incorrectly stopped early for declaring futility. And our type 1 error rate is 5%, and that's pretty good. But now under H0, we're incorrectly stopping early for efficacy in 3% of cases. But here's our big efficiency gain: 40% of studies are stopping early for futility at the interim analysis. So we're less likely to go the full distance there. So what's the take-home message? Take-home message is that we've got a plan for our possible adaptations in advance and account for them in the analysis. So rather than just going, I don't want to run a fixed design study, I'm going to run an adaptive design, we actually need to plan in advance when we're designing the study, what kind of adaptations are possible and how is that going to affect kind of the operational characteristics of our study, the type 1 error and the power and everything, and then account for that when we're analyzing the data as well. So that was a kind of detailed look into one particular very simple type of adaptive design. I'm going to go kind of quickly through some other adaptive designs that are used with a few examples, which might inspire you to realize that they could be used in your work. So this is kind of similar to stopping early. This is called the promising zone approach, which is to say that we do an interim analysis where we're not going to stop for definitive efficacy or futility at that interim analysis. And it doesn't look like we've got a good chance of getting to that point by the end of the planned study as well, but the results are kind of looking promising. We might decide to continue beyond the original planned sample size. So this is distinct from blinded sample size re-estimation. So blinded sample size re-estimation is where you've made some assumptions in your sample size calculation about event rates or standard deviations of your outcome variable. And at halfway, you might go, oh, it looks like we underestimated the variance, so we need to bump up our sample size. You can do that in a blinded fashion, and that's perfectly fine from a stats point of view. This is based on unblinded data because you need to compare the groups. And it's based on the conditional power. So this is the probability that you're going to get a significant result by the end of the study. And so what you would typically do is kind of pre-specify some range of conditional powers where you say, if with our current sample size, we've got between like a 50 to 80% chance of reaching the significance, we might expand our sample size so that we can push that up to 80 or 90%. Rather than just sticking with what we've got. So there are kind of more details here. I think these slides will go up on the Stats Central website, so you can click on those links there. And just a couple of examples of these in real life. So the COVA study was looking at a specific type of steroid hormone in COVID-19 treatment. They had an initial sample size of 310. They did an interim analysis at halfway, and it had promising results. So they set a new target sample size of 375. So from 310 to 375. What ended up happening is that they stopped at 238 due to a low recruitment rate, unfortunately. But they were able to show that this treatment was effective in reducing the rate of respiratory failure or early death in COVID-19. So that's good. And this one, I'm not even going to try to say that, but it was looking at a type 2 diabetes drug in stroke, recruiting patients within 9 hours of a stroke and testing if this drug helped with their neurological symptoms. They had an initial sample size of 528. They did an interim analysis at 320, with the possibility of increasing the sample size up to 650. Again, if things were looking kind of promising. They too, unfortunately, had to stop early at 350 due to COVID-19 disruptions and funding constraints. So this is not an unusual thing to happen. But again, I guess the point I'm making is that you can plan these things in advance. And you might make the decision to increase your sample size as you go. Again, that's going to have kind of budgetary consequences as well, so you got to think about that. Seamless designs are pretty neat. In clinical trials, especially, this is like where you combine a phase 2 study with a phase 3 study. So phase 2 study might have like an initial assessment of efficacy, maybe with a surrogate endpoint that's quicker to collect than your main clinical endpoint. And you might have a bunch of treatments, and you might just choose a couple of them that you want to test further. Some that are looking promising in this initial phase. And then phase 3 is like the definitive assessment of efficacy using like a longer term clinical endpoint. And a seamless design basically, puts these into one study. And this can be operationally seamless, which is where you kind of have no gap between the phases where you have to, start a study again and reintroduce sites and train people up and get ethics approval and everything like that, because it's all covered under one protocol. Or it can be inferentially seamless, which is where the same participants are included in both phases. So again, this phase 2 might be short term, and then you can follow up those same participants if things are looking promising for those longer term outcomes. And you can do analyses that take that into account. And just a couple of examples. This TV practical study was one about trying to cut down the burden of treatments for tuberculosis. So second line treatment for that used to involve up to 20 tablets a day for up to 20 months, which you can imagine is quite burdensome. But they wanted to test 3 new regimens that had fewer tablets a day and that only required 4 months of treatment. And as I said, for that, they had a short-term outcome. So they just looked at, culture conversion in the blood at 8 weeks after taking this drug. And then of those 3 possible treatments, after phase 2, they decided on just one of them that they would then carry into phase 3. And that had a primary outcome at 72 weeks, which was a clinical endpoint of, death or tuberculosis recurrence. This was inferentially and operationally seamless. So the patients were recruited to all 4 arms while they were still making that stage 2 decision. And those same patients were still followed up to 72 weeks, which is good. And it ended up showing that this selected regimen that made it through phase 2 was safer and more effective than the standard of care. So that's pretty good. This second one is testing a monoclonal antibody in COVID-19. So they weren't sure what kind of dose you should give people. So in phase 2, they tested a high dose and a low dose and a placebo. It looked like the high dose was working all right. So that went into phase 3. Both stages used the same outcome of 28-day mortality, but the study design was so that they would have 95% chance of selecting the best arm in stage 1, and then 85% power to find a significant difference in stage 2. So again, this is combining those 2 trials into one trial to save yourself time and effort, but taking into account when you are designing it in the first place. Enrichment studies is the idea that while you're accumulating the data, you might identify subgroups of patients who the treatment is working particularly well in, and you might then decide to, target recruitment towards those subgroups of patients so that you get more of them in the study. And so that those people who might benefit from the treatment are more likely to receive the treatment. So this is particularly kind of useful in cancer studies where like biomarkers can identify meaningful tumor subtypes. So if you test people's genes before they start the treatment or the tumor genotypes, you might identify that this treatment is working in patients with a particular mutation in their tumor, and then you can target recruitment towards those patients as you go. I've included a couple of examples here. They're both pretty similar. So they're both about kind of targeted therapy for cancer, and in both cases, participants were genotyped at the start of the study, randomly assigned to treatments, but then as treatments showed more efficacy in certain subgroups of patients, then patients with those same characteristics were kind of targeted to receive those treatments kind of further on in the study. Response adaptive randomization is one where instead of keeping equal allocation to the different treatments, you can change that ratio during the study so that more participants are assigned to treatments that look like they've got better outcomes. And there's kind of a level beyond that, which is covariate adjusted response adaptive randomization. So that might change the ratio differently for different participants. So if your treatment looks like it's working especially well in older participants, you might have a 3-to-1 ratio for older people and a 2-to-1 ratio for younger people, for example. A couple of kind of caveats to keep in mind. So this is obviously beneficial in the sense that participants are kind of more likely to get a treatment that's going to work. Caveats are that equal randomization is statistically the most efficient for comparing groups. So if you go away from equal randomization, you might end up lowering your power and ending up needing more participants in total anyway. And you also need to be careful about time trends and adapting this randomization ratio too early or too often. So if you remember back to our simulated studies, they could be quite volatile early on. And if you're adapting your randomization ratios kind of too early, you might be your results might be too sensitive to those early kind of fluctuations. So a couple of examples. NTB was kind of a follow-on trial from the one we talked about earlier, where they were trying to find shorter regimens for tuberculosis. And in that case, yeah, the randomization began balanced, but they updated it monthly so that more participants were assigned to arms that looked like they had greater interim treatment effects. And a kind of special case of this is that if a treatment is looking like it's really not working, you can drop its randomization ratio down to 0, which is essentially dropping the treatment out of the study altogether. And BATTLE was a targeted therapy for lung cancer trial. They used equal allocation for the first 80 patients, and then they used this CARA idea of changing the randomization depending on your tumor subtype to determine which targeted treatments you might get based on the results that had been seen in similar patients so far. Finally, as an example, this is kind of like the granddaddy of adaptive designs, adaptive platform trials. So this is like you set up a platform where you're going to do trials from, and the trial itself is kind of continuously updating and it's perpetual. It can keep going for as long as you like, combining a kind of master protocol with an adaptive study design. So a master protocol is like kind of the overarching, well, I've called it a core protocol here. So it's the kind of overarching or generic features of the study. So, you know, what disease you're studying, what endpoints you're measuring, what kind of principles you're going to use for making adaptive decisions. And then you've got these appendices for particular domains. So this might be a patient subgroup. So, you know, patients with a particularly severe disease might be involved in one domain, or it might be treatment. So there might be different types of treatments that you're going to compare. And you have an appendix that, you know, gives the specific details of these particular domains. So, you know, you might have inclusion and exclusion criteria specific for certain patient subgroups, certain treatment comparisons. You might need to have the details of, you know, how you administer those treatments, and interaction with other domains. So if you've got different types of treatments that could be given at the same time, you want to have some rules about how people might be randomized to both of those domains if they meet the inclusion and exclusion criteria. So this can incorporate you know, a huge number of adaptive aspects. So treatment selection, you know, adding and dropping treatment arms as the results come in, enrichment, so targeting patient subgroups, and adaptive randomization. And the main thing is that this is a lot of work. This needs careful planning and ongoing monitoring. Kind of one of the most famous adaptive platform trials is actually run out of Australia, REMAP CAP. That's a platform trial for community-acquired pneumonia. And they did pretty well because they had kind of designed it, I think in 2017 or 18, to be able to handle a global pandemic of respiratory disease. So during COVID especially, they had a lot of patients come on board, and you know, up to now they've, you know, recruited over 13,000 patients. And those patients, because they can be randomized multiple times to different treatments, they've had over 23,000 randomizations. They've investigated 66 interventions in 18 different domains. So these are examples of domains. So testing different antibiotic therapies in severe pneumonia, different durations of this therapy for severe pneumonia, different antiviral therapies for COVID, convalescent plasma for COVID. And these domains kind of might be closed if the question's no longer valid. Maybe if another study's already shown that this treatment works or doesn't work, you can stop. Or they have statistical triggers. So if you show that the longer macrolide therapies obviously works really well, you might stop randomizing people to that particular domain. But the overall kind of platform trial goes on as new treatments and questions come up. So quickly, I'm running pretty close to time here. Just some advice for planning an adaptive study. Number one thing is you've got to pre-specify the adaptations that could occur, and thinking about the timing of that is important. So we're not only interested in the primary outcome when we run a clinical study. You want to also know things about safety. So if something's looking really efficacious, but you haven't followed people long enough to know if it's safe, then maybe you don't want to be stopping the study at that time point. And like I showed earlier, you need to kind of think about how the adaptations are going to affect your observed data, define your decision rules to kind of control your error rates, account for your adaptations in the analysis. And this is all kind of usually done by kind of simulating what your study would look like under various scenarios. And from that, you can kind of get expectations of the time and the cost efficiencies. I did that first example kind of under frequentist paradigm, which is where you kind of consider what the data could look like under various scenarios. This is tricky to do with really highly adaptive studies because p-values are difficult to calculate in those situations. An alternative is a Bayesian approach, and these are kind of pretty commonly used in adaptive studies. They produce posterior probabilities that are conditional on what you observe, not what you might have observed. But I guess the one thing is that if you're making a decision, and you are, because you're deciding whether to adapt your study or not based on posterior probabilities, you still want to maximize the chances of making the right decision under various scenarios. And so in these studies, these decision rules, even though they're based on Bayesian posterior probabilities, they're often kind of defined and evaluated in terms of like their frequentist properties like power and type 1 error rates. And that involves simulations again. So where to next? Adaptive designs can add efficiency, but they can make things more complicated. Operations, costs, your data management, your logistics can be more complicated. Asking for money to run a perpetual study might be difficult. And your stats is difficult as well. And I know the title said clinical and other studies, and I've only talked about clinical studies. The reason for that is this is kind of most applicable, I guess, to clinical studies because typically you've got kind of ongoing recruitment and you can stop kind of midway through and make a decision before you decide to go, instead of like a I don't know, like an engineering study where you've got all your samples at the start. But yeah, it could be used in any area where a research question is sequential, where you can kind of say, I've got a bunch of questions I want to ask, but I need to know the answer to the first one before I ask the second one. But the same statistical principles apply. You need to think about what your data is going to look like as you make those adaptations. So where to next? Speak to a statistician early. Stat Central have free study design advice and a whole bunch of other services that I'm sure you know about and that were showed at the start of the presentation. Again, you can click on these to go to the website. Clinical research unit where I'm based. We've got statisticians and health economists who work closely with the Kirby and the George Institute, who have a bit of experience with adaptive designs. We've also got other kind of helpful things we can provide. So protocol and budget development, ethics submission help, legal advice, data management, and so on. And we've got a community on Teams that has just started up, which is kind of like a place where people can connect and collaborate with clinical studies. So thanks very much, and apologies for not leaving very much time for questions. I check the chat.
Question.
Yep.
So are these sort of techniques commonly accepted, or do you think people have problems publishing paper? Like, what's the sort of pushback from reviewers with these sort of adaptive designs?
Yep. So the question was about kind of how well accepted these designs are among reviewers mainly. Yeah. So I would say that they're not super uncommon anymore. So I think they're pretty well accepted. Like the FDA, for example, has specific guidelines for adaptive designs. So they accept, drug registrations for drugs that have gone through designs like this. So they're happy with it. And they're, famously pretty careful about stuff like that. I haven't personally tried to submit a paper based on an adaptive design, but I get the impression, especially during COVID, because a lot of studies were run as adaptive designs during that time. Even in like the big medical journals, they're pretty comfortable with it now. Yeah, as long as you are careful about how you design it.
I've got a question. Is there like any software that will do this for people?
Yeah, question was, is there any software that will do this for people? So like the design?
Yeah.
Yeah, there are, depending on which type you're talking about. So there are group sequential designs is not a new thing, like stopping early, that's not new. So there are a bunch of software that'll do that for you. All the other stuff is generally simulation based. I know there are some software that do it. Berry consultants in the US are big in this area, and they have kind of custom software that does that kind of stuff. I don't think it's cheap though. And I spoke to someone who has used that software, and they actually said, because there's such a huge range of adaptations and things you need to consider, it's hard for any software to cover all those possibilities. And he found that it actually ended up being faster for him just to code the simulations himself. So yeah, it's very simulation based, and it might be very kind of custom to your particular scenario. But maybe some software could give you like a ballpark to start with. Something in the chat.
No.
Ah,